So we talked about DNA becoming RNA, becoming protein in ribosomes. And we didn't mention the fact that where is the ribosomes are actually functioning in order to create a protein. So inside you have the DNA, and then you have the RNA, and the RNA is going to move through many different processes that is going to, from pre-mRNA, is going to become a mature RNA, and then it's going to move out through the pores to the cytoplasm, and then it is going to meet a free ribosome, and then it is going to become a polypeptide. But wait, is it becoming a polypeptide specifically here? So in this video I would like to talk about the endoplasmic reticulum, and specifically about the rough endoplasmic reticulum. The rough endoplasmic reticulum is an extension of the outer membrane of the nucleus, and it is striated with many different ribosomes. The fact that it is striated by so many ribosomes is because ribosomes that are going to be attached to the rough endoplasmic reticulum in order to create and to translate the mRNA into polypeptide inside the rough endoplasmic reticulum. But why do we need to create polypeptide inside the rough endoplasmic reticulum? So I mentioned that you have the mRNA, which is going to go out through the nuclear pore, and then it is going to meet a free ribosome. Now, this is the point when we have two main things that can occur. One, the polypeptide is going to just become a polypeptide, the mRNA is going to be translated inside the free ribosome inside the cytoplasm, and this polypeptide, this protein, is going to usually stay inside the cell in order to be and take functions inside the cell. But we can also take this ribosome with the mRNA, and this mRNA is going to have something called a signaling sequence, a signaling zone. And the signaling zone is basically going to signal different functions and parts around inside the cell. It is going to signal that this polypeptide specifically needs to be extracted out of the cell. And in order to do it, in order to extract out things outside of the cell, we need to pass the plasma membrane of the cell, and we need to do something called exocytosis. And in order to do it, we need to pack the polypeptide inside the vesicle, and in order to do it, we need to translate the polypeptide inside the endoplasmic reticulum, specifically the rough endoplasmic reticulum. So this structure is going to go, and the ribosome is going to bind to the surface of the endoplasmic reticulum, the rough endoplasmic reticulum. The polypeptide is going to continue to translate inside the rough endoplasmic reticulum, and then you are going to see this bud of vesicle going outside and either going to the Golgi apparatus for further modifications we are going to study in the future, or is going to leave the cell through the plasma membrane through exocytosis outside of the cell. So we are going to see what is exocytosis, what is the Golgi apparatus, and why it is important that we are going to create the polypeptide that is needed to go outside of the cell, needed to be extracted outside of the cell in the endoplasmic reticulum in future lessons. But for now, I would like to say that because we need to take the thing outside of the cell, and we need to pass the plasma membrane of the cell, imagine this is part of the plasma membrane and this is the cell, we are going to create a vesicle that is basically going to fuse together with the plasma membrane, and the protein is going to be extracted out of the cell. In order to do it, we have to have a vesicle, a plasma membrane around the protein. The protein will be inside. So we do it using the endoplasmic reticulum. So if I'm going to sum this video up very quickly, we said that one, both pathways have a common ancestor, which is the DNA created pre-mRNA, created the mature RNA, going out of the pore, and always meeting a free ribosome outside of the nucleus. Only when meeting the free ribosome, it is going to pick two paths. If the protein is going to stay inside the cell, is going to just continue the translation inside the cell, inside the cytoplasm. But if we need to extrude it out using exocytosis and pack it into a vesicle, it is going to be using a signal peptide, a signal area. It is going to go and bound to the rough endoplasmic reticulum. It is going to continue the translation inside, and then this vesicle is going to bud off, and it's going to either go to extrude or going to further modification in the Golgi apparatus. And we're going to study everything in the future.